Hello, my name is Heidy Elkhaligy. I am a master's student in the biology department at Florida International University. I will present my poster titled Comparative Analysis of Structural Features in Slims from Eukaryotes, Bacteria, and Viruses, with importance for host-pathogen interactions. Cellular functions are regulated by protein-protein interactions, and alterations in the protein interaction network can lead to disease. This poster will focus on the protein interactions facilitated by short linear motifs, or SLIMs for short. SLIMs are short amino acid patterns ranging from 3 to 10 amino acids. SLIMs are known to perform vital interactions in eukaryotic cells, but sequence-based predictions of SLIMs have a high false positive rate. In eukaryotes, SLIMs found in a conserved protein region implies that they are important across homologs. Due to random mutational processes, proteins from pathogens such as bacteria and viruses can evolve and acquire SLIMs that mimic the proteins of their host in a process called molecular mimicry. By mimicking host SLIMs, pathogens can hijack cellular functions and alter the signaling pathway of the host cell. Pathogenic SLIMs tend to vary in their sequence conservation. Although most eukaryotic SLIMs interactions are transient, some pathogenic SLIMs were discovered to have high affinity toward the interacting protein partner. In eukaryotes, SLIMs are often found in intrinsically disordered protein regions that generally lack a specific secondary structure. That's why disordered is one of the main attributes used to detect functional SLIMs. However, the overall disorder content in the eukaryotic proteomes is higher than in other pathogenic proteomes. The differences in disorder content between eukaryotes, bacteria, and viruses raise the question of whether SLIMs in pathogens are indeed disordered as eukaryotes, or could they have different structural properties? Finally, are other structural features universal for SLIMs? Currently, the best database that contains all the experimentally verified SLIMs is the Eukaryotic Linear Motif Database, or ELM database, which contains around 4,000 experimentally verified instances from approximately 300 motifs in proteins from eukaryotes, bacteria, and viruses, divided into six functional categories: cleavage, degradation, docking, ligand binding, post-translation modification, and targeting motifs. Our objective is to use sequence-based structural predictions to compare structural features of the true positive SLIMs in the ELM database to explore the differences or similarities between taxonomic groups like eukaryotes, bacteria, and viruses, and between different ELM types. All the true positive SLIMs and their protein-containing sequences were extracted from the ELM database. Then we used sequence-based predictors such as IUPRED2A to predict the disorder of each amino acid in an instance. In our analysis, we used the default cutoff value of 0.5 used by the predictor and the zero point 0.4 cutoff value based on the previous literature to determine whether the amino acid is disordered or not. In addition, we used NetSurf-P2 predictor to predict the relative solvent accessibility and secondary structure of each amino acid in an instance. We calculated scores that enabled us to complete our analysis, such as percent disorder, percent accessibility, and percent of predicted coil amino acids per instance. Moreover, we calculated the average disorder score per instance, which is the mean IUPRED disorder score, or MIDS, and the average coil confidence score, which is the mean coil confidence score, or MCCS. For data analysis and visualization, we used Python, SciPy, Matplotlib, Seaborn modules, and SQL. Our analysis of disorder percent per instance revealed a clear split between the completely disordered and completely ordered SLIMs based on both cutoff values 0.5 and 0.4, where viral SLIMs in blue are often less disordered than SLIMs from eukaryotes in gray and bacteria in green, as you can see in figures A and B. Furthermore, all taxonomic groups showed a higher percent of instances in the completely accessible and completely coiled categories as in figures C and D. These results suggest an interplay between order and disorder, with accessibility and coiled may impact the SLIM functionality in all taxonomic groups. Based on our analysis of the taxonomic groups, disorder, MIDS, and coil confidence and CCS percentages, we think that coil confidence score may improve the identification of functional SLIMs as shown in the lower figure. The same analysis, when colored by ELM type, demonstrated that most viral SLIMs involved in post-translation modification, or MOD instances colored in light blue, have lower mean IUPRED disorder score or MIDS values than eukaryotes. However, Bacterial slims showed a flip between being ordered when using the long disorder prediction to being disordered when using short disorder prediction. This suggests that post-translation modification sites in bacteria are found in short disordered regions that lie within structural domains rather than in long disordered regions in bacterial proteins. The MCCS of viral post-translation modifications support the MIDS results by having lower values with great variability, suggesting that some modification sites are ordered, not disordered or coiled. The average disorder analysis highlighted that SLIMs in viruses proteins are more ordered and appear to be less coiled than host proteins. It's plausible that viral molecular mimicry is mediated through a more limited set of conformations than in the host. This may be due to different functional constraints imposed on viruses during evolution that can enhance their interaction with host proteins, while interactions between host proteins are often under tight regulation that may include on and off states. We also investigated differences and similarities between the shared motifs between viruses and eukaryotes and between bacteria and eukaryotes. Furthermore, we explored specific types of motifs that emphasize that there are similarities and differences between SLIMs on the motif level. If you are interested in knowing more about the studied motifs, you can scan the QR code found below to find out our published manuscript in Pathogens. Thank you for listening.